Okay, so folks, some announcements first. So welcome to week 7 of 16. We're almost at the halfway point. So you guys, we're going to do it. We're going to do this. There's one handout. This is for our new unit, Unit 5, on microbial growth. So we'll be starting this today. Also, folks, those of you that returned your lecture exam 1, version A, you'll recall there was a mistake on the key. So I recorrected them. Most of you had the correct answer. So I added, because the exam was out of 200 points, the correction was I added 2 points to your total score if you had the correct answer there. A few folks didn't have the correct answer even after I corrected it. So some of you, maybe there's no change, but most of you got an additional 2 points, so well done. Let me see here. Right, so what we'll do today, folks, is we'll finish metabolism. The last topic on our PowerPoint B was the nitrogen cycle, and we're going to simplify it and use the handout that we passed out last time on the nitrogen cycle. I'll just do kind of a skeleton of it on the board. But folks, be aware that on lecture exam 2, I love to ask application questions on the nitrogen cycle, and so we'll go over those. And the applications, like why we care, are down at the bottom of the handout. Okay, and then we will start our next unit, Unit 5, Microbial Growth. I have a whole bunch of them, you guys. In fact, there's the nitrogen cycle and also this really nice diagram that Selena, a previous micro student, she brought this in from A&P, and it's a nice schematic of aerobic respiration in human cells. So many people said it was easier to understand than the crazy bacteria that we put up here. So folks, are there folks here that need the handouts? We'll just pass them all on. This is from last time, you guys, the nitrogen cycle and the aerobic respiration in a human cell. So again, folks, what we're going to do is just do a skeleton of the nitrogen cycle, and I'll kind of quiz you as we go. So folks, is molecular nitrogen, 2 nitrogen atoms with that strong triple covalent bond, is this a usable form of nitrogen? Meaning, can organisms use it to make nitrogen-containing organic molecules like amino acids or proteins or nucleic acids? No, that's not usable, right? So, in the process called nitrogen fixation, the molecular nitrogen will be converted to what? Ammonia, good. Now, is ammonia a usable form of nitrogen? Meaning, can organisms use it to make amino acids, proteins, nucleic acid? Yes, it is, right? But this takes a really complex enzyme complex. What is the enzyme complex called? Nitrogenase, good. And the process, folks, is called what? What's this process called? Nitrogen fixation. We're fixing the nitrogen into a usable form. Can you name 2 bacteria we've studied in lab that can carry out nitrogen fixation? Awesome. Cyanobacteria? Such as nostoc. And you guys, is nitrogenase inactivated by molecular oxygen? Yeah, the phrase is it's oxygen sensitive. So how does cyanobacteria protect nitrogenase from molecular oxygen that's being made in the photosynthetic cells? Heterocytes, right? Different cells, right? So the heterocytes, there's no oxygenic photosynthesis. It's an anaerobic environment, so it protects the nitrogenase from inactivation. So it's within the heterocytes that nitrogen fixation is occurring. Awesome, you guys. What's another bacterium? Good. Rhizobium, and Rhizobium are soil microbes, right? Rhizobium also can make nitrogenase. How does a rhizobium protect the nitrogenase from molecular oxygen? What's the strategy? Yeah, right. Remember, they form a symbiotic relationship with which group of plants? Legumes. What are legumes? Peas, bean, clover, alfalfa, lupins, right? So the legumes form the root nodules, and what's the environment like inside the root nodules? Anaerobic, right? So with the rhizobium live in the root nodules, and they carry out nitrogen fixation. Awesome, folks. Okay. Now let's presume that maybe we do have some legumes growing in our soil. They've been colonized by Rhizobium, right? The Rhizobium are making ammonia, sharing it with the plant, but also some is kind of leaching into the surrounding soil. There are other soil bacteria that can take the ammonia and convert it into nitrates. And this process is called nitrification. Nitrification. And we need not memorize the names of the bacteria, folks, that carry out nitrification, but nitrates are also used by plants, are also usable as a source of nitrogen to make nitrogen-containing organic molecules. Okay, so folks, if we are farmers, right, and we want our crops to grow and make lots of food for us, as farmers, do we want nitrogen fixation and nitrification to occur in our fields? Yeah, right? And I just make a happy plant. Plants love nitrogen fixation and nitrification because, again, the plants can use the ammonia and the nitrates as a source of nitrogen to make proteins and nucleic acids to grow, right? But, folks, there's another side of our cycle here. And we'll see that this the third stage of the cycle is where we take our usable nitrates and we're converting them to what? Yeah, molecular nitrogen. So we're going from usable nitrogen to what? Not usable nitrogen. So this process, folks, we'll put it in red. Well, is called denitrification. And again, it's carried out by microbes. And the microbes, folks, will carry out denitrification when the soil becomes anaerobic. So denitrification, folks, this is an example of when bacteria in the soil switch to anaerobic respiration. Remember, as microbiologists, when we see respiration, we know there's an electron transport chain involved. The anaerobic is telling us what about the final electron acceptor? It's not oxygen, right? And it makes sense this is going to happen under anaerobic conditions. So when would we have anaerobic conditions in the soil? This is usually when it gets waterlogged, right? Saturated with water. Waterlogged, meaning that the soil is saturated with lots and lots of water. And the reason is oxygen does not diffuse well through water, right? And so when might we get waterlogged soils? Yeah, big rainstorm, right? Rainstorm, flooding. And if you're not a good farmer, if you over-irrigate your fields, what is irrigation? You're watering your fields, right? And what if you're watering too much, right? Okay, so over-irrigation. So again, folks, because now the soil is anaerobic, could the bacteria carry out aerobic respiration? Nope. So those that can will switch to anaerobic respiration. Do you remember we said one of the alternate terminal electron acceptors in anaerobic respiration was nitrate? You remember that? And what happens when it accepts all those electrons? It gets reduced back to molecular nitrogen. Okay, so folks, let's take a look at our handout then. Yeah.
Can I just ask a quick question?
Yeah.
So you said for nitrogenase that was the enzyme process, correct?
The enzyme complex?
Enzyme complex.
Yeah, complex.
And then,
Yeah.
Would you say for N fixation?
So if I asked you which process does nitrogenase catalyze, it's nitrogen fixation. So the enzyme is nitrogenase. The process, the reaction is nitrogen fixation. You are welcome. So folks, on the nitrogen cycle handout, down at the bottom, there's applications of the nitrogen cycle. So let's take a look at that, because I love to ask these types of questions on lecture exam too. So we're looking specifically, folks, at denitrification. We would argue as farmers, do we want denitrification to occur? You could argue not, because it's stealing usable usable nitrogen from our plants, right? They're not going to grow as well. So certainly, as farmers or gardeners, we don't want denitrification to occur because, again, it's stealing usable nitrogen from our plants so they can't grow as well. But, folks, there are a couple of examples when denitrification is actually beneficial. And so number 2 down there, folks, it says, what if you're working in a wastewater or a sewage treatment plant? And, folks, what happens is with wastewater sewage treatment, the water is treated, and then it's dumped into our rivers, our coastal waters, right? So, folks, would you want a lot of usable nitrogen in the treated wastewater that you're going to dump into the rivers and coastal waters? You wouldn't want a lot of usable nitrogen in there because who, living in the water, could use it to multiply to really high levels? Algae or cyanobacteria, right? So if you're working in sewage or wastewater treatment, you want denitrification to occur so that you will get rid of usable nitrogen in the treated water. So when you dump it into the rivers or the coastal waters, you won't get those harmful algal blooms. And those harmful algal blooms, folks, some of the algae make toxins. Harmful algal blooms can also include overgrowth by toxin-producing cyanobacteria. And those toxins can actually cause harm to humans, to marine mammals. And furthermore, if you have this big overgrowth of these harmful algae or cyanobacteria, eventually they die. And then other microbes in the water use aerobic respiration, and they degrade the dead cells, and they use up all the oxygen. So you can end up with these aquatic habitats that are depleted of oxygen, and then like fish can't grow, you end up with these dead zones, right? So one way we can try to prevent those harmful algal blooms is to make sure that our wastewater, our sewage water, has very, very little usable nitrogen in it. And one way we can do that is to encourage denitrification. The third example, folks, is, again, if we're farmers and we're being responsible farmers, one mistake we might make is adding too much artificial nitrate-containing fertilizers to our fields, right? Because we know that nitrates help our plants grow. So because we want our plants to grow fast and produce a lot of food, we might add artificial nitrate fertilizers. But this can be a problem because if we add excess nitrate fertilizers, if there's a rainstorm or if we over-irrigate, the runoff water is going to have a lot of what in it? Nitrates, right? And it's the same thing as with the sewage or wastewater. It runs into our streams and rivers and eventually into the coastal regions and can contribute to what? Harmful algal blooms, right? So one way that responsible farmers can decrease the chance of excess nitrates from their fields running into our waterways is to use bioreactors. And this is really cool, you guys. Down at CSU Monterey, they have a bioreactor experiment going on, and they involve their undergraduates and their graduate students. And the simplest kind of bioreactor, folks, is you could literally just dig a hole in the ground. So this would be the soil. And then you usually add some kind of substrate, like wood chips. So these could be, these are big wood chips, right? And then what you're going to add is soil containing soil microbes. Many of whom we know can carry out anaerobic respiration. So then what you do, if these are your fields, and this is the runoff water from your fields with excess nitrates, you first run it through your bioreactor. So this is your bioreactor, right? It's going to fill up with water. So is that going to be aerobic or anaerobic, guys? This is the water, right? So you're going to have anaerobic conditions. What are the soil bacteria going to do? They're going to switch to anaerobic respiration. What are they going to use as their terminal electron acceptors? What are you trying to get rid of? You're trying to get rid of the nitrates, right? So using anaerobic respiration, the nitrates are going to get blown off as what? Right? Molecular nitrogen is a gas, right? So by running the water through your bioreactor, the bacteria release the nitrates is unusable molecular nitrogen. And then folks, the water then that exit is going to be very low in nitrates. And then this is going to go into the streams, our rivers, into our coastal waters. And why is that beneficial, you guys, to get rid of those nitrates? Yeah, so it's going to prevent those harmful algal blooms, right? And so this is just, again, folks, taking a problem that we humans kind of made by using these artificial fertilizers and using microbes as kind of an ecological solution, right? Very low tech, doesn't cost a lot of money, right? So this is really good when the farmers are interested in protecting our water quality. And just one last thing, folks, the other thing I'm really interested in is using nitrogen fixing like Rhizobium and legumes. Instead of adding artificial fertilizers to our garden, I try to plant, beans and peas and lentils. And so we'll get the Rhizobium making the ammonia, enriching the soil naturally, right? And then the soil bacteria make the nitrates. So when you're growing legumes, you never end up over-fertilizing. You never have an excess of nitrate. So it's a much more ecological way, right, to introduce usable nitrogen into your soils for your plants. Yeah. Okay. So folks, this handout then, again, I love to ask these kind of application questions on lecture exam too. So I would suggest you. Folks, for denitrification, know when it's considered detrimental, right? Know when it's considered beneficial, okay? And also, folks, for farmers, know that for farmers wanting their plants to grow quickly, that farmers want to promote nitrogen fixation and nitrification, okay? So with that, folks, we're going to close our metabolism unit. And we're going to move to the next unit, which is microbial growth. And there was a handout today, folks. It's kind of a combination supplemental lecture notes or kind of key points from lecture. And then towards the end, folks, it's the study guide. So it's a combination supplemental notes and study guide. Okay. And this is a relatively light unit. We should finish by the start of Thursday's lecture, right? So this is our Unit 5 microbial growth. And folks, we'll be focused on the growth of bacteria. And when we're talking about microbial growth, it's often different than, say, if you're talking about growth in, say, ANP, right? Because we're not talking about a multicellular organism. So when we're talking about growth, we're not talking about an increase in size or an increase in weight, because that would be so hard with these little tiny bacteria. So when we're talking about microbial growth, we're talking about the growth in population. So an increase in the number of cells in a population, right? So it's a little bit different than how we would maybe talk about the growth of an individual human. So folks, these are the topics we're going to hit in this unit. So we're going to talk about how bacteria reproduce asexually in a process called binary fission. And then we'll show you what's called a standard bacterial growth curve, the 4 phases of bacterial growth. And then we'll talk about factors influencing growth. So we'll talk about nutrients, carbon and energy sources. We'll talk about the impact of the presence or absence of molecular oxygen. We'll talk about temperature, pH, and the osmotic environment of cells to see how that influences their growth. So first of all, folks, again, we're focused on bacteria. So if we were to watch bacteria, we would see that a single cell is going to grow bigger, copy its chromosomal DNA, and then do what? Split in two. So this is binary fission. It's asexual, right? There's just one parent. And the time it takes for one cell to split into 2 cells is called the doubling time or the generation time. Okay, the doubling time or the generation time. And the doubling or generation time is encoded in the chromosomal DNA. So some bacteria, like E. coli, under ideal conditions, has a generation time of only 20 minutes. So that means every 20 minutes, what's happening to the population size? It's doubling, right? But in contrast, some bacteria, say our Mycobacterium tuberculosis, Mycobacterium leprae, they might have doubling times of weeks, right? It takes them weeks until one cell can divide into two. So it's hardwired in the chromosomal DNA. This electron micrograph, folks, it's been artificially colored after it was taken, but this is just a cartoon of a bacterium undergoing the process of what? What's this process called? Binary fission, right? What do you think this orange material is? The chromosome, right? So folks, in the next unit in microbial genetics, the first topic we're going to tackle is how do the bacteria make copies of the chromosomal DNA. It's a pretty amazing process. Now, early bacteriologists, they wanted to be able to run lab experiments to plot bacterial growth in a lab. So this is an artificial setting, folks. So this would be an experiment where you take a flask of sterile medium, say broth. You're going to inoculate it with some living bacteria, keep it warm, and then maybe every hour or so take a sample from your broth culture and determine the number of cells per mil. So you're determining the number of living cells per mil of broth. And a fancy term, you guys, for living cells, they're called viable cells. So if you see viable, we're just going to presume that just means they're living, they're not dead. Okay. So again, folks, this would be, we'd have a sterile flask with broth in it, right? We're going to inoculate our bacteria, and then we'll incubate them, keep them warm. And then we're going to take samples, again, and then we're going to graph the number of viable cells per mil. So here, the x-axis, you guys, will be time. And then we're going to start out with, folks, when folks first started running these experiments, the y-axis initially was just simply the number of viable cells, living cells, per mil. Okay? And the problem was, like initially in the experiment, it started out okay, right? Because you take a sample of cells, and maybe every hour, maybe this is like E. coli, right? And initially you're okay, you're going along, it seems like nothing's happening, but then suddenly you have an explosion of cell numbers. And so your data points just, you'd have to have graph paper like 3 feet long, right? So the answer to this was that the microbiologists switched to using semi-log paper to do their graphing. So let me just change this, you guys. So here on semi-log paper, the x scale, the x-axis is still arithmetic scale, but the y-axis is a log scale. So y is the log of the number of living cells per mil, right? And so this means you can use just a regular piece of graph paper. And in doing this experiment, folks, again, we want to remember it's in the lab. It's a closed system. We're not adding more nutrients. We're not removing waste products, right? So in the lab, what you'll get is a graph that has 4 distinct phases. So we want to talk about those phases, their names, what's happening, right? So if I connect my data points here, we'll see 4 distinct phases, and this is the so-called standard bacterial growth curve. And again, folks, it's used, we use semi-log paper. And again, folks, I could ask you, what's the x-axis? It's time. It's an arithmetic scale. The y-axis is the log power of 10 of the number of living cells per mil. Okay. So let's go ahead then and look at this standard bacterial growth curve. And folks, what I usually do on our lecture exam too, I'll put a diagram of the standard bacterial growth curve, and of course I'm going to white out the label of the x-axis. I'll white out the y-axis label, and I'll white out the names of the phases, right? So you'll want to be able to label the x and y axis. You want to be able to label the 4 phases, and then I'll ask you questions about what's going on. Okay. So first, folks, let's talk about the 4 phases, the names of the 4 phases. So initially, folks, it's like there's no change in the number of living cells. This is called the lag phase. And it makes sense, folks, that initially when we just first introduce our bacteria into this new environment, it takes them a while to become adjusted to their new environment. It's going to take them a while to transport nutrients into the cell, right? They have to synthesize, transport proteins, they have to transport nutrients into the cells. And then they have to start biosynthesis. They have to start making proteins. They have to copy their chromosomal DNA. They have to make RNA, right? So this takes time. So for a while, it looks like they're not doing anything during this lag phase, but they're getting prepared to start dividing like crazy, okay? And then, boom, all of a sudden, we have massive increase in cell numbers. This is when the bacteria are dividing as fast as they can, given the environmental conditions. This is called the log or exponential phase, and again, it's when the bacteria are dividing as fast as they can, given the environmental conditions they have. Now, what I'd like you to remember, folks, is during the log or exponential phase, again, this is the maximal growth given the particular environment. It is when cells are most sensitive to antimicrobials. So if it's a bacterial pathogen, this is when the cells are most sensitive to antibiotics, antiseptics, and disinfectants, right? It's the best time to perform gram stains. Why is that? If you want to avoid false gram-negative results, when should we be gram staining our gram-positive bacteria? During the log or exponential phase, right? Because it's then that they have the thickest, continuous layer of peptidoglycan, right? There's not holes in it. So the young gram-positive bacteria in the log or exponential phase, they're going to trap that crystal violet iodine inside, and they won't decolorize. And it's also during the log or exponential phase that you can calculate the doubling time or generation time for that particular bacterium. Okay. Now, again, folks, remember, this is a closed system, right? It's in a flask. So what's happening to nutrients as a population size increases? Nutrients are going down, right? What's happening to waste product, toxic metabolic waste products? They're going up, right? So is the environment kind of degrading, deteriorating? Yeah, getting kind of nasty in there, right? So we will then reach the third phase called stationary phase of growth. And in the stationary phase of growth, the new cells that are arising from binary fission are equal to the number of cells dying. So it's like zero population growth, right? There's overall no change in the number of living cells. And then a few years ago, one of your colleagues was studying ecology and asked a great question. And so he said, for that particular environment, is this the carrying capacity, the maximal population size that that environment can sustain? And the answer is yes, that's brilliant, right? This is the maximum population of cells that that particular environment can sustain. You guys probably know that we're worried here on planet Earth that are we reaching our carrying capacity, right? Where the Earth can't sustain a larger population size. That's always a huge worry for us. Furthermore, folks, in the stationary phase, because the microbes aren't growing as rapidly, they're less resistant to antimicrobials. So they're not going to be as sensitive to antibiotics. This is one of the reasons why bacterial pathogens living in biofilms are much more resistant to antibiotics. Not only are the layers of the biofilm a diffusion barrier against antibiotics, the bacteria in the lower layers are in the stationary phase of growth, so they're naturally more resistant to antibiotics, which is a big problem, right, with biofilms. Okay. Oh, and also, folks, let's pretend you're using an endospore-forming bacterium in your experiment. And remember, folks, endospores are formed by members of the genus Bacillus, like Bacillus anthracis that we looked at in lab, and also by the genus Clostridium. So, folks, what is the function of an endospore? Do you remember? There you go. Spot on, right? The endospores are there to protect a copy of the chromosomal DNA, to protect the organism when environmental conditions get horrible, right? So, you guys, what would be your guess as we move from the log phase, the late log phase into stationary phase? Do you think we're going to start seeing endospores produced maybe in this transition? Yeah, makes sense, right? The environment's getting bad. You know, the parent bacteria are probably going to die. So it's probably in late log that we start seeing endospores produced. folks, what's going to happen as time continues? What's going to happen, do you think, to the concentration of endospores? Going to keep going up, right? Because things are getting really nasty now. Okay. So again, folks, in our closed system, right, getting to late stationary phase, what's happened to all the nutrients? Probably gone, right? What's happening to waste concentrations? Really high, right? This is a nasty environment. So what's going to happen in the fourth phase? The number of cells dying, or the number of dead cells, exceeds the number of living cells, right? So this is called which phase? The death or decline phase, right? And again, folks, we would predict that we would see lots of endospores if we were doing cultures on Bacillus or Clostridium. If they were gram-positive bacteria, folks, in the death or decline phase, do you think we'd see lots of false gram-negatives? Yeah, and why is that again? The peptidoglycan's breaking down. Exactly. Can't trap that crystal violet iodine inside. Good job, folks. Okay, and let me just see if I've hit all the topics here, folks, to make sure. So again, you guys, make sure there's going to be at least, maybe 4 questions on this diagram. You want to be able to label it and then tell me what's happening in the different phases. So for sure, we're going to see that on the lecture exam too. Okay. Questions, folks? Okay. All right, so we looked at our standard bacterial growth curve, but now we want to investigate, well, how could we influence generation time? How could we influence microbial growth? If we want our microbes to grow better, what do we have to provide? If we want to inhibit the growth of unwanted microbes, how could we change conditions to inhibit the growth of unwanted microbes? So one thing we always need to think about if we're trying to grow microbes is what is their source of carbon and what is their source of energy. So there's 4, we could say, energy, nutritional classifications based on an organism's energy and carbon sources. So folks, the only 2 categories you need to know for lecture exam 2 are the photoautotrophs and chemo heterotrophs. Okay? So you guys, with photoautotrophs, the reason I'm standing over here is, oh, sugar, I forgot to plug in the microphone. Oh, no. Oh, no. Oh. Okay, gosh darn it. Okay, sorry. Forgot to turn on the microphone. So folks, with photoautotrophs, what is the energy source that photoautotrophs use? Light. Photo means light, right? So photoautotrophs use light as their energy source. Autotroph means self-feeder. So autotroph is telling us what is their carbon source. And so what's the carbon source that photoautotrophs use to make, say, organic molecules? Carbon dioxide, right? Good old carbon dioxide from the air. So you guys, for photoautotrophs, what's their energy source? The energy source is light. Energy source is light. And what is their carbon source to build organic molecules? Yeah, CO2. Awesome. Can you name 3 groups of 3 groups of organisms we've discussed in lab at least that are photoautotrophs? So algae, cyanobacteria, plants. Good. And furthermore, folks, do you remember that primitive type of photosynthesis called anoxygenic photosynthesis? Yeah, those bacteria would also be considered photoautotrophs because they're using light energy and CO2 as a carbon source. They're just not pumping out O2, but they're still considered photoautotrophs. Good. And folks, the other group you want to remember are the chemo heterotrophs. So folks, what is the energy source for chemo heterotrophs? Preformed organic molecules. And what's their source of carbon? Preformed organic molecules, right? So folks, let's walk through some organisms. Kingdom Animalia are members of Kingdom Animalia photoautotrophs or chemo heterotrophs? Chemo heterotrophs, good. Kingdom Fungi, are they photoautotrophs or chemo heterotrophs? Chemo heterotrophs, all of them are chemo heterotrophs. Good, you guys. Amongst our protists, what about I'm stumbling here because I always think of euglena. That's always like okay. Most of the animal-like protists, most of the protozoa are what? Chemo heterotrophs. Good. And euglena, right? That's always the wonderful, goofy one. Good job. So folks, do you think there are members of domain bacteria that are chemo heterotrophs? Yes, right? Are there members of domain bacteria that are photoautotrophs? Yes, good. Okay, all right, so we got that. And it's unfortunate, folks, we just don't have enough time to get into the photoheterotrophs nor the chemoautotrophs. So the only two you're going to see on lecture exam 2 are which ones? Photoautotrophs and chemoheterotrophs, good. Oh, down here, folks, I had a little note to myself because I always forget this. So you will often hear, especially like pathogenic microbes that invade another organism and cause harm. Often they're what we call fastidious microbes. And fastidious means they can't hardly make any of their own organic molecules. It's almost like you have to spoon feed all of their organic molecules preformed. And again, folks, this helps us understand, why do they invade humans? We're all these wonderful preformed organic molecules, right? And we'll see in lab the reason this is important. It's important to know that a lot of human pathogens are fastidious, because if we take a sample from our infected patient and we want to try to grow the microbe in the lab, we have to supply them with really rich media with all these preformed organic molecules, vitamins and amino acids and the precursors of nucleic acids, because they can't make any by themselves. And if we don't give them all of these preformed organic molecules, they won't grow in the lab, and we might come up with a false negative. Yeah. So again, when we start talking about, it's chapter 10, unit 10 in lab, microbial media, we'll be talking about enrichment media to grow fastidious pathogens. Good, okay. All right, now you guys, this looks really boring, but this is such a cool topic. So let me go to one of the slides that has some visuals on it, so it won't be quite as boring. All right. So folks, we just talked about how the carbon source and energy source can influence whether our microbes will be able to grow or not. So now we're going to look at the influence of molecular oxygen. And this totally amazed me, folks, because I always presumed all organisms are like us. All organisms require molecular oxygen, right? I just thought, well, of course, everybody's like us, you know. But it turns out molecular oxygen living in an aerobic environment, it is really hazardous. It's really hazardous, so we want to explore this a little bit more. So folks, let's take kind of an evolutionary approach to this. All right, you guys, so like 3.6, 3.8 billion years ago, when the first cells evolved, right, about 3.6 to 3.8 billion years ago, what was Earth's atmosphere like? Anaerobic, right? So it was an anaerobic atmosphere. So it makes sense, folks, that the first early cells, they would carry out anaerobic metabolism to make ATP. So for example, fermentation, right? And then eventually, when those porphyrin rings evolved and we had evolution of cytochromes, which permitted evolution of electron transport chains, we had anaerobic respiration, right? But in addition to the evolution of cytochromes, porphyrin rings permitted evolution of what other really wonderful thing? Yeah. Remember, you guys, the porphyrin rings permitted evolution of bacterial chlorophyll. It'll only take a minor mutations to get chlorophyll A. So chlorophyll A evolves, and what does chlorophyll A permit? Oxygenic photosynthesis. Now, why was that a big event? How did that change? How did it slowly change Earth's atmosphere? It took a while. Yeah, but slowly, right? Slowly, we have an accumulation of molecular oxygen. And this blows me away, but this accumulation of the molecular oxygen, the aerobic environment, it probably caused a massive extinction event. And I'm like, what are you talking about, right? How is that possible? And the reason is, folks, is that when microbes or cells, organisms, us too, are living in aerobic environments, oxygen can interact with components of our cells, and we can get these toxic, what we'll call reactive oxygen intermediates, right? So we're going to have formation of toxic, poisonous, what we'll call reactive oxygen intermediates, or ROIs. And I know you guys, abbreviations are maddening and I use them. So we call them ROIs, for reactive oxygen intermediates. Different authors will call them different things. But I would like you to know, folks, too, of these toxic reactive oxygen intermediates, which will be generated when organisms are living in aerobic environments. And so one of the really nasty ones is called superoxide. The superoxide anion or radical. So an example of some of the free radicals you've probably heard about in the press, like there's research points to that free radicals, when we're exposed to free radicals, it contributes to aging, it increases our risk for cancer, right? And the reason is that these free radicals cause all kinds of damage to cell membranes, to proteins, to DNA and RNA, right? Nasty, nasty things. And the superoxide anion, folks, it's a molecule of molecular oxygen, but it has an extra electron. That's an extra electron. And it carries a net negative charge. And again, you guys, so damaging. Oh, my goodness. And then another one that organisms living in aerobic environments, another one of these toxic ROIs that they're going to be exposed to is hydrogen peroxide. H2O2. Not as toxic as the superoxide anion, but still can do a lot of damage. And so it's like, wow, that could have wiped out life on Earth. But we want to remember, folks, remember how we said that waterlogged soil will be anaerobic, right? So thank heavens on Earth there would have been these little anaerobic ecological niches where there was either no oxygen or very low concentrations of oxygen. And these were almost like little nurseries, right? Little protective nurseries. And so you can imagine wherever there was waterlogged soil, or maybe, I don't know, the bottom of the ocean, right? These are anaerobic areas. So anaerobic ecological niches. And we could argue that this permitted a protective environment for natural selection to occur, right? So the selection for mutants that made enzymes that could break down these reactive oxygen intermediates, right? So it provides protection so that we have natural selection for mutants which make enzymes to detoxify, make unharmful, to detoxify the reactive oxygen intermediates. And indeed, folks, organisms like ourselves that live in an aerobic environment and organisms like us that have to have aerobic respiration, right, to make enough ATP, we have inherited the genes for these protective enzymes. Otherwise, folks, we could not survive in our aerobic environment. So what we want to do is take a look at two of the enzymes that would help break down these reactive oxygen intermediates. And of course, I would like you to know the names of those two protective enzymes because you've inherited them. You make them yourselves, right? So the enzyme, folks, that's going to break down the superoxide anion is called, let's see, I should do this better. So the superoxide anion will be detoxified by an enzyme called superoxide dismutase. Superoxide dismutase. And sadly, because we use an abbreviation for that, we call it SOD. Right? Not very elegant, but superoxide dismutase is going to help detoxify the superoxide anion. and it's going to break it down into hydrogen peroxide. Now you might say, wait, hydrogen peroxide is also toxic, but it's far less toxic than the superoxide anion, right? So what about an enzyme that's going to help detoxify the hydrogen peroxide? And there's two enzymes that evolved to take care of this. We'll talk about the one that we made. So for hydrogen peroxide, folks, the enzyme that we make that will detoxify is called catalase. Okay, so detoxified by catalase. And I'll put in parentheses, you guys, or another enzyme called peroxidase. All right. Now that we have a little background, and we will come back to this slide, you guys. Let me go back to that initial text-heavy slide, because it actually has the chemical reactions that superoxide dismutase and catalase catalyze. So let me go back here, and folks, you wouldn't have to remember these chemical reactions, but I just want you to see just kind of two-step process. So folks, here's that really toxic superoxide anion. So superoxide dismutase is going to break it down into the less toxic hydrogen peroxide and will release molecular oxygen. Now in your cells make superoxide dismutase, right? And your cells also make catalase. And what catalase will do is take that hydrogen peroxide and break it down into water and molecular oxygen. So now in this two-step process, you guys, we've taken this really nasty, highly toxic superoxide radical, and we've broken it down into water and molecular oxygen. Okay? So in lab, we're gonna run a benchtop fast test to see if your microbes make catalase, and this is so fun. I'm easily entertained. And this will be one of the biochemical metabolic tests we run to identify your unknown bacteria. Real simple test. Get a clean microscope slide, you put a drop of 3% hydrogen peroxide, and then you harvest your bacteria, say from your agar plates, and mix them in to the hydrogen peroxide. If you see bubbles, what is that telling you? Your microbes are making catalase, because remember, folks, in the catalase reaction, the hydrogen peroxide is broken down in water and molecular oxygen. Is this a gas? Yeah. So that's what's forming the gas bubbles here, right? This is a molecular oxygen that's being released as the catalase breaks down hydrogen peroxide. In the old days, folks, we would use hydrogen peroxide as an antiseptic. So like if you had a cut or a wound, you'd pour the hydrogen peroxide in it, and what would you see? Bubbles, right? Because your cells make catalase, yeah? Nowadays, they're kind of trying to discourage people from using hydrogen peroxide. I love it, you guys. I'm all scarred and broken down anyway. As I'm pouring that stuff in there, it's bubbling away. I'm like, yeah, take that, microbes, right? Because it's antimicrobial. And the other really cool thing is, you guys, and I think this is something, I don't know, it's like I'm not a human healthcare provider, so I should be careful. But we were taught, like with literally dirty, penetrating wounds, hydrogen peroxide is really good because the molecular oxygen helps physically bubble out dirt and debris, right? So I still like it, but you're not supposed to use it anywhere because it does cause damage to your cells as well, right? But I love it when I have a wound, pour that in there. I'm like, die bacteria. Anyway, okay. So we'll be running this in the micro lab, okay? So why do we care? Well, we can run experiments in the lab, and we're going to run this experiment in the lab to explore our microbes' relationship to molecular oxygen. Can they grow in the presence of molecular oxygen? Are they killed in the presence of molecular oxygen? So let's take a look, folks. This again will be another diagram on lecture exam 2, and I'm going to white out all the labels, right? And I'm going to ask you questions. These cartoons, folks, again, this is similar to the experiment we'll run in lab. The little dots represent where the microbes are growing. Again, you can see they're growing in different areas of these tubes. Now the tube, the medium, is a special type of medium called thioglycolate. And the reason we use thioglycolate is it will reduce molecular oxygen and help create an anaerobic environment. Okay. So folks, and again, we're going to do this in lab. We're going to use this media called thioglycolate. And what it does, it reduces molecular oxygen and it creates an anaerobic environment. So with our tubes of thioglycolate medium, we'll have this lovely oxygen gradient. So this will be our tube, right? This will be our thioglycolate. So we're going to have a high O2 concentration up here at the air medium interface, right? And what's happening to the oxygen concentration, you guys, as we're going down through the tube? Yeah, we're going to have like no O2. So we've got an O2 gradient. And the presumption is it's totally anaerobic in the butt, right? The butt is the bottom of the tube, and it's going to be aerobic at the medium air interface. And what you'll do is we'll be using different microbes. You're going to use your needle, and you're going to stab straight down and straight back up. You don't want to shake it, right? You don't want to shake the tube or shake your loop or needle so that you're depositing the microbes all throughout this O2 gradient. And then we're going to incubate them. And after incubation, again, we take them out of the incubator really carefully. We don't want to shake them. We're going to look at the pattern of growth. Okay, so some microbes like the Pseudomonas, and this Pseudomonas, you guys already know, it causes really severe respiratory tract infections in our cystic fibrosis patients. Nasty, nasty guy. So Pseudomonas is going to grow up at the top where oxygen is present. Now, why do you think Pseudomonas is only growing up at the top where the O2 is present? Yes, they like us, have to use aerobic respiration to make their ATP, right? So are they going to die if no O2 is present? Yeah. That's right. They can't make any ATP, right? So folks, microbes that require molecular oxygen because they have to use aerobic respiration are called obligate or strict aerobes. Now, here's a follow-up question. Do you think these obligate or strict aerobes like Pseudomonas, do you think they're going to make those protective enzymes, superoxide dismutase and catalase? They have to, right? Because they're using oxygen in aerobic respiration. They're generating so many of those toxic reactive oxygen intermediates. They have to make those protective enzymes. Otherwise, what would happen to them? They're going to die, right? Okay. Now, at the opposite extreme, folks, is microbes such as members of the genus Clostridium. And this is important to remember, you guys. Clostridium are what we call strict or obligate anaerobes. What does that mean? They're killed in the presence of oxygen, right? Why? Why are they killed in the presence of oxygen? What can't they make? They can't make, what's protecting us, you guys? Our superoxide dismutase and catalase. Can any of the members of genus Clostridium make superoxide dismutase or catalase? No. So why can't they grow in the presence of oxygen? Those reactive oxygen intermediates kill them, right? Where can they grow? Only in anaerobic environments, right? So you guys, how are they making their ATP anaerobically? Fermentation, or maybe anaerobic respiration, right? As long as there's no O2 involved, they're okay. Hey, does this make sense, folks? Stricter obligate aerobes make at least the superoxide dismutase and catalase. Obligate anaerobes can't make any protective enzymes, right? Does that make sense? Okay. Now, the next one here, folks, I love this one. This is our good friend E. coli. And we can see that E. coli is growing throughout our thioglycolate. It's growing aerobically, it's growing anaerobically. But do you notice a difference in cell concentration? Yeah, a lot more at the top than at the bottom. And this is a reflection of a microbe that can use aerobic respiration if O2 is present. But if there's no O2, they can switch to fermentation or anaerobic respiration. Okay? So in which of those three processes will the microbes make the most ATP? Aerobic respiration, right? I mean, right, hypothetical 38 ATP per glucose, right? So they're going to be dividing like crazy up here aerobically, right, making all that ATP. let's presume, you guys, just for this example, that there's no alternate terminal electron acceptor for anaerobic respiration. Let us presume that down here in the bottom, E. coli have to carry out which process? Fermentation, right? And how many ATP per glucose? 2, right? So you would argue E. coli is dividing like, what, 20 times faster up here aerobically than down here, right? They're making almost 20 times more ATP up here than down here. Are they killed aerobically? Nope, right? So they aren't killed aerobically, they can grow anaerobically. Such organisms are called facultative anaerobes. Because they're using aerobic respiration, do you think they make protective enzymes like superoxide dismutase and cylce? Yes, they are. They are, right? If you're carrying out aerobic respiration, you have to make those protective enzymes. Okay. All right, folks. I have one more after this one, but see, this is a example of that big family of bacteria we call the lactic acid bacteria. Okay. So the genus Streptococcus is a member of the lactic acid bacteria. Now, do you see? Are they growing aerobically? Yeah. Are they growing anaerobically? Yeah. And I wish they'd been a little bit more careful, but there should be equal cell concentrations, folks. Aerobically, anaerobically, the same cell concentration. And the reason is the genus Streptococcus, they never evolved the ability, this was the story I heard, to make porphyrin rings. So they never evolved the ability to make cytochromes. So what can't they make? Electron transport chains, right? So they can't carry out aerobic respiration. They can't carry out anaerobic respiration. What do they do to make their ATP? Fermentation, right? And specifically lactic acid fermentation, that's why they're called lactic acid bacteria. So only 2 ATP per glucose, right? So they're not going to have really high cell concentrations, right? Because they're only making 2 ATP per glucose. Do you think they're making some protective enzymes? Yeah, and it turns out the members of the genus Streptococcus, since they're not using aerobic respiration, they get away with an equivalent of catalase called peroxidase, right? So they're not using O2, right? So they can get away with just an enzyme that will help detoxify hydrogen peroxide. And I'll just throw this in, you guys, don't worry about this for lecture exam 2. This is for lab. If we did the catalase test on a Streptococcus, they won't make bubbles because the reason is peroxidase, it breaks down hydrogen peroxide, but it doesn't release molecular oxygen, right? So again, that's just for lab, you guys, and maybe just don't even worry about it. We'll talk about it later in lab when we do our metabolic tests. Okay, okie dokie. And then, folks, there is, I've got another example here, but before we leave this slide, folks, remember how I said Clostridium or Obligate Anaerobes, right? So why is it when we talk about like tetanus infections, the classic example, you guys, is you step on a rusty nail and you get infected with Clostridium tetani. But that rusty nail is in what kind of environment? Aerobic. So and I just told you Clostridium are killed by aerobic environments. How is it that the Clostridium are on that rusty nail? Or how is it they're in the soil, there's air in the soil? What do they make that protects them? Endospores, right? So you guys, with the Clostridium, the endospores are protected under aerobic conditions. And this is why you can become infected with Clostridium and you're living aerobically, right? So the endospores protect the Clostridia under aerobic conditions. And so that's important. That's important to know. They can only grow under anaerobic conditions, but those endospores whose job is to protect them, right, the endospores will protect them from being killed by aerobic conditions. Yeah, good. And then, folks, the last, I want to give you a 5th example because with Thanksgiving coming up, this becomes really important. There's another classification of microbes. And these guys are called microaerophiles, little air lovers. So these micro, aero, we inoculated them into our thioglycolate. Are they going to grow up at the top? No. Are they going to grow at the bottom? No. They're going to grow somewhere in between. So these little guys, my understanding is, and I'll give you an example. This is a bacterial pathogen. It's a really important foodborne pathogen called Campylobacter. They're microaerophiles. My understanding is they can only make really low concentrations of protective enzymes. So they can't live, atmospheric air, you guys, is about, it's about 21% O2. So they don't have enough protective enzymes to live aerobically up here. Right? They make a little bit, so they can grow somewhere where the oxygen concentration is lower than 21%, right? Now, why am I bringing this up? Because this was a real mystery. Campylobacter, you guys, they're just presumed, okay, let me back up here. So transmission is fecal-oral, right? They grow in animals, they grow in poultry. So poultry, chicken, turkeys, ducks, right? And when the chickens and turkeys are slaughtered, folks, just presume there's a fine frosting of fecal bacteria on the outside of the carcass. And this is true probably for all raw meat. I mean, we just have to be realistic about it. So the reason this was kind of a, was a mystery, and folks, this is supposed to be a turkey. This is a turkey because we have Thanksgiving coming up, raw. So this is a raw turkey, right? So people classically are infected through improperly cooked poultry. You know, chickens and turkeys are a big one, right? Because traditionally what you do, you stuff the cavity, and often the insides don't get heated high enough to kill the fecal bacteria, right? So remember, folks, when you're working with raw poultry, just presume there's this frosting of fecal bacteria on it, including Campylobacter. Presume all raw poultry has Campylobacter contamination. So the mystery was, how do the Campylobacter, right? It's on the outside, right? The fecal pathogens are on the outside. How does the Campylobacter survive on the surface of the carcass, right? Because that's 21% molecular oxygen. So I think it was a team of German researchers. They figured it out. It's brilliant, guys. What do the Campylobacter do? They make a biofilm, right? So they're going to form a biofilm, right? So here's our biofilm, folks. And guess what? It's an O2 diffusion barrier. So in a weird sort of way, you guys, it's sort of like our thioglycolate tubes. You'll have high O2 up here, and then next to your raw chicken or turkey, right? High O2 here, low O2 here. Does that make sense? In the deeper layers. So where are they going to grow? In the place that's just right. So again, folks, to me, it's really fascinating to see how microbes evolve strategies to live in these different environments, right? And here, a real practical application is these microaerophilic Campylobacter form the biofilm, and then they can continue to grow and replicate in your raw poultry. And again, with Thanksgiving coming up, we're going to have all kinds of Thanksgiving food microbe, food infection examples coming up. Okay, all right. This is just an example, folks. If we wanted to say, let's say we were trying to grow Clostridium in the lab, we'd have to somehow create an anaerobic environment for them. In the old days, they'd use candle jars. So they'd put their cultures in the jar and light a candle, put it inside it, and seal the lid. And that would use up the oxygen. They now have these gas packs, these catalysts that will reduce all the molecular oxygen to water. And then in a nicer lab, a nicer diagnostic lab, they'd have an anaerobic chamber here, so everything's done anaerobically, right? But we don't have this, so we don't grow anything anaerobically in our we don't work with any stricter obligate anaerobes in our micro lab, right? Because we don't have the right environments to prevent them from dying. So you guys, this is just, we talked about how oxygen can influence microbial growth. Just really quickly, folks, we're going to talk about temperature. So this was an experiment where E. coli was growing at 3 different temperatures, 22 degrees Celsius, 30 and 37 degrees Celsius. So you guys, what do you see as the temperature increases? What's happening to the number of colonies and the size of colonies? They're increasing, right? So if we were to plot microbial growth, the growth rate, so we could say maybe the doubling times against temperature, we would see as the temperature increases, what's happening to the growth rate. It's increasing, right? It's getting faster and faster. This makes sense, you guys, because temperature is the average kinetic energy, the average speed with which molecules are moving in a cell. So if we have an enzyme and a substrate at low temperatures, what's happening? Very few enzyme substrate collisions, but now let's increase temperature, things are moving faster and faster, right? So now at a higher temperature, we're going to have more enzyme substrate collisions, right? So the cells can grow and divide faster until, what the heck is happening right there? We get to this optimal growth rate, optimal temperature, and then suddenly we have a sudden decrease in growth rate. Why? Denaturation, right? Now things are moving so fast as molecules collide, proteins are unfolding. We even get DNA and RNA denaturing. I know we don't talk about that much, but they do, right? So usually microbes then have, through natural selection, they evolve to have their maximum growth rate at the temperature of the environment in which they've been growing for generations. Okay. And folks, you don't have to remember the temperature ranges, but we classify microbes based on their optimal temperature. So we classify them as cold-loving or heat-loving. So we have psychrophiles that can grow well at refrigerator temperatures. Mesophiles, you guys, meso means moderate temperature. So most microbial pathogens of humans are considered mesophiles because they grow best at about what temperature? 37 degrees Celsius, right? So most of the microbial pathogens growing in your human patients, they're probably going to grow best at around 37 degrees Celsius. There's always exceptions, but most of them are what we call mesophiles. And then there are heat lovers, right? Thermophiles, and then hypothermophiles. So folks, 100 degrees Celsius, this is where water is boiling, right? Isn't that crazy? So thermophiles and hypothermophiles, these are ones we could find growing around hot geysers, hot springs, maybe where there's active volcanoes. They'd be living close to the environment of where that the lava is actually flowing. It's pretty remarkable. So folks, with these guys, what I'd like you to remember is that most microbial pathogens that live in humans are going to be what? Mesophiles, right, growing around 37. And then, folks, there's another really important bacterial pathogen that I want you to know about because we have it here in California. Okay. Oh, this is just cool, you guys. Just as an aside, I wouldn't ask this on the exam, but sometimes in the Sierra Nevadas, you'll see snow that looks like it's blood-stained, and you're like, what happened? This is horrible, right? But these are psychrophilic algae that can grow in the snow, and they have a beautiful red pigment. So you can impress your friends and family if you go up in the snow and you see, oh my God, there was a massacre here. You go, no, it's just photosynthetic algae, psychrophiles, Anyway, okay. But this is the one, folks, that I really want to develop because we have this pathogen here in California and it's caused some kind of heartbreaks. And this is Listeria monocytogenes. And this is really an interesting bacterial pathogen. Okay, so the pathogen, folks, is Listeria monocytogenes. This is a bacterial pathogen. I think named after Joseph Lister, the person that developed surgical anesthetic. Monocytogenes because it can grow in our mononuclear cells. It can grow in our monocytes and macrophages, which is bad news. And this little guy can be transmitted fecal orally, which whenever we have outbreaks, they never say that. They never tell you it's from fecal contamination, right? They never say that. Anyway, so what's interesting about this little guy is that it can grow inside humans. And thus, how would we classify it, you guys? It would therefore act as a what? As a mesophile, right? But what is wild is that it can switch to becoming a psychrophile because it can also grow in refrigerators. And refrigerators, you guys, most of them are like around 4 degrees Celsius. So if it's growing in the refrigerator, folks, how would we classify it? So 4 degrees Celsius is here. So it can switch to being a psychrophile. And indeed, if you're working, say, in a public health lab, and you get like a fecal sample in, or you get some water or food that you think has been contaminated with feces, and you're screening for Listeria, you stick the sample in the fridge, because that will slow down most of the other contaminating bacteria who continues to grow. Listeria. So you actually do a cold enrichment for them, right? And then you'll plate them to look for the presence of the Listeria. Who cares? All right. Well, these are really bad news, folks, for pregnant women and anybody immunocompromised. Because in pregnant women, immunocompromised. So we'll do the pregnant women first, folks. So often where we get infected, frequently it's from raw milk products. I shouldn't say frequent because I'm going to get yelled at, but sometimes found in raw milk products. What's raw milk? Right, raw milk is unpasteurized milk. It hasn't been heated to kill any milk-borne pathogens. And the reason, folks, it can be found in raw milk is because cows can become infected. So cows infected, and what happens is you can get fecal contamination of the milk. And you might think, no, there's no way. But guys, I worked on a raw milk dairy. Wow. You know, after we milk, I would clean the filter. The milk runs from the cows and milking machines through pipes to a cooling tank, right? And there's a filter there. And thank heavens there's a filter there because I would clean it and there's like pieces of cow feces in the filter, right? So, and if you've ever worked on a dairy, you know why. When it's rainy and slushy and the cows are, anyway, we won't go there. The cows are infected, right? So you can have fecal contamination of their milk. But this is really bad news, you guys. Fecally contaminated milk. But remember, you guys, how we said monocytogenes? These little guys have evolved to grow in monocytes and macrophages, including the monocytes and macrophages of cattle. And do you think that milk, mammalian milk, has white blood cells in it? Yeah, there's white blood cells in milk, right? So the cows, when they get infected, we can have infected monocytes, macrophages. In the milk, right? And so if the infected monocytes or macrophages are in the milk and they're carrying the Listeria, right? That's another way the milk gets contaminated. Not a problem if the milk is pasteurized. Big problem if it's not. So if a woman eats a raw milk product that's contaminated with Listeria, she gets infected, and the Listeria, this is a heartbreak, you guys. So if the woman ingests the Listeria contaminated milk, right? It replicates, Listeria replicates, and it crosses the placenta. Yeah, we call it transplacental transfer. So now who's going to get infected? The baby. And this is usually devastating. So this is called transplacental transfer. And we're always worried because the developing baby is at really high risk. So what can happen is you can end up with miscarriage. This is what happened several years ago. There was an outbreak of Listeria from raw milk product, and it caused a series of miscarriages in women, right? And if the baby survives, so we have infection of the baby in utero. So if the baby survives, often what you'll end up with is what's called neonatal newborn sepsis and meningitis. So the bacteria is growing in the bloodstream of the baby and invading the central nervous system. And that can kill the baby or cause permanent neurological damage. So this can cause death or neurological damage. So this is devastating, guys, neurological damage. Now, in addition, recently, it's been, Listeria has been found to contaminate fruits and vegetables. And there was a big outbreak in Colorado linked to melons. And nowhere did I see in the reports that they said this is fecal contamination, right? I don't know why. It's like the public should know this is fecal contamination. So with regard to milk, you guys, if you have pregnant patients or you're pregnant or family or friends are pregnant and traditionally they drink raw milk, just tell them while you're pregnant, just please switch to pasteurized milk products, including cheeses, buttermilk, kefir, any kind of milk product while you're pregnant. Go for pasteurized, right? Because we have this in California. We've had outbreaks and, big heartbreak to lose your baby or have your baby be sick from it. And again, folks, keep your ears peeled for outbreaks in plant products like fruits or vegetables, right? And again, it's fecal contamination. And it might be from animals or it could be from humans, yeah? And the other group you guys be really careful about is your immunocompromised patients, because this can cause really serious disease in the immunocompromised. So your HIV/AIDS patients, transplant recipients, cancer patients, I would just tell them stay away from raw milk products, right? Always go with pasteurized, yeah. Okay, so that's just my little blurb there, you guys. But just wild how this Listeria can switch from being mesophile to a psychrophile and back again. Just crazy. All right, almost finished, you guys. Oh, and again, folks, this is just an example of hypothermophiles. This is in the hot springs, the geysers of Yellowstone. And they found bacteria and archaea that can live really close. They're obviously not living in the boiling water, but they live in the environments where the hot water is draining off. Really hot temperatures, right, up around 70 degrees Celsius. That would burn us. And then, folks, I know we just have 2 minutes, but we'll just introduce this. So, folks, we want to look at how pH and osmotic pressure can be used to control the growth of microbes. And our application is food preservation, okay? So with regard to pH, folks, organisms that can live at acidic pH, and acidic pH is less than 7, they're often called acidophiles. And I would say, folks, are lactic acid bacteria, right? The lactic acid bacteria, if we use them in fermentation of milk, they can drop the pH from pH 7 to pH 4, right? They're fine living at pH 4. Right, 1,000-fold increase in hydrogen ions. That's going to inhibit the growth of a lot of spoilage microbes, right? So lactic acid fermentation of milk, of vegetables helps preserve them. There is one bacterium, you guys, that can actually live in our stomach, and for many years we said it's not possible for a microbe to live in our stomach, but this is the potential pathogen called Helicobacter pylori. It turns out it has a buffering system. It produces urease. It produces ammonia. So it buffers the stomach pH so it can live in the mucosal epithelial cells there. Neutrophiles are going to grow best where? Around neutral pH, right? So most of the microbial human pathogens are going to be neutrophiles. And then alkalinophiles are those that grow best at alkaline pH. And just one example, folks, they often use is the bacterium that causes the diarrheal disease cholera. It can grow happily at pH 9. Okay? So we'll stop there, you guys, and then we'll talk about osmotic pressure, and then we'll show some examples of how controlling the pH and osmotic pressure can help us preserve foods. Okay. Oh, yeah, you should. No problem. You are welcome. Thank you. Great. So is Gabi here or Julie? Okay, let's see here. What are we doing? Oh, I should stop this, huh?
Is this the only one that you handed out today?
Yes, that's the only new one.
Okay. These are just extra ones that I had. Excellent.
Thank you, thank you, thank you. I appreciate it. I appreciate it. Thank you.